Fibrous proteins, such as collagen, are most abundant in the body. Fibrous proteins are important for us. For example, without collagen, we can't even make the bones. Fibrous proteins have several characteristics. The key characteristics of fibrous proteins include long, narrow rock-like structure and low water solubility. Today, we will focus on three topics: collagen, elastin, and keratin. Let's look at the first part. Collagen is the most abundant protein in the body. 3 alpha chains of tropocollagen form a right-handed triple helix. The alpha chain consists of glycine XY repeats. Here XY typically represent prolyl and hydroxyprolyl, or prolyl and hydroxylysine. Collagen makeup is superfamily, and they include 25 collagen types. For this lecture, we will focus on 5 types, except type 1 and type 4 that are made up of 2 alpha-1 chains and 1 alpha-2 chains. Type 2, 3, and 5 are made up of 3 alpha-1 chains. Type 1 and 3 are particularly important because defects with this collagen cause several collagen-related diseases. Collagen is produced by osteoblasts to make up the bone, by chondroblasts to make up the cartilage, and fibroblasts to make up extracellular matrix. Similar to transferrin, synthesis of collagen goes through Golgi before it is secreted. But different from transferrin, collagen is modified before and after secretion. Inside pro-alpha chain is hydroxylated at prolyl and lysine residues. 3 pro-alpha chains form triple helix. At this stage, this molecule is called procollagen. After secretion, the ends of procollagens are cleaved. Now, this is called tropocollagen. Tropocollagen associate each other to form collagen fibril, and fibrils aggregates to form collagen fiber. Here is a collagen fibril. Each of these is a tropocollagen. During formation of collagen fibril, cross-linking occurs between tropocollagen molecules. Each of these is a cross-link. This is a summary. Synthesis of collagen goes through procollagen, tropocollagen, collagen fibril, and then to collagen fiber. Here is a more detailed description of synthesis of collagen. Inside the pro-alpha chains are hydroxylated at prolyl and lysine residues by 2 enzymes, prolyl hydroxylase and lysyl hydroxylase. In transferrin, O-link glycosylation occurs in serine and threonine, but in collagen, it occurs in hydroxylysine. 3 pro-alpha chains form triple helix. Now, collagen is secreted as procollagen. After secretion, loose termini are removed by peptidases. Now the triple helical molecule is called tropocollagen. tropocollagen associates with each other in staggered parallel array to form collagen fibril. Cross-linking occurs between L-lysine and lysine or between L-lysine and hydroxylysine among tropocollagen molecules. This is important for tensile strength of collagen. Cross-linking is catalyzed by lysyl oxidase. Hydroxylation at the proline and lysine is catalyzed by prolyl hydroxylase and lysyl hydroxylase respectively. For example, here is a proline. After hydroxylation, it becomes hydroxyproline. This enzyme requires vitamin C as a coenzyme. Prolyl hydroxylase is important for triple helix. In particular, prolyl hydroxylase is important for interchain hydrogen bond formation and formation of the stable triple helix. Hydroxylysine is important for glycosylation of collagen. Either galactose or glucosyl-galactose is added to hydroxylysine in collagen, contributing to collagen's tensile strength. Hydroxylysine also involved in cross-linking. Vitamin C deficiency impairs interchain hydrogen bond formation and the formation of stable triple helix. Vitamin C deficiency also causes failure of glycosylation. These together leads to scurvy. Early symptoms of scurvy include malaise and lethargy. After a month, patients developed shortness of breath, bone pain, skin changes with roughness, easy bruising, and petechia, and gum disease. Cross-linking is catalyzed by lysyl oxidase. Here is a lysine residue on collagen. After this reaction, amino group is removed and aldehyde group is generated. This reaction converts lysine to L-lysine. Cross-linking occurs between L-lysine and lysine residues. This enzyme requires copper as a cofactor and vitamin B6 as a coenzyme. On the other side of the story, collagen is degraded by collagenase. Here are collagen-related diseases. Two major diseases are Osteogenesis Imperfecta and Ehlers-Danlos Syndrome. Here is an example of a mild osteogenesis imperfecta with blue sclera. In this patient, sclera is thinner than normal because of defects in collagen. As a result, the underlying veins become visible. This disorder is caused by mutation in either alpha-1 or alpha-2 chains of type 1 collagen. Mutation causes misfolding of procollagen. These are examples of more severe form of osteogenesis imperfecta. On the left panel, the fetus has fracture. On the right panel, crumble long bones. Here, long bones look like being crushed together to produce irregularity. And beaded ribs. Appearance of large beads around ribs. Ehlers-Danlos Syndrome features hyperelasticity of the skin, hyperextensive joints, aortic aneurysm, rupture of colon, and skin hemorrhage. All these are because of defects in type 1, 3, or 5 collagen, caused by mutation in either alpha-1 or alpha-2 chains of type 1, 3, or 5 collagen. It might also be caused by mutation in lysyl hydroxylase or procollagen peptidase. Airport syndrome arises due to defects in collagen present in basement membrane caused by mutation in type IV collagen. Goodpaster syndrome arises due to autoimmune response. Menkes disease features depigmented and steely hair or kinked hair. It is an X-linked recessive disorder caused by copper deficiency. Lack of copper impairs lysyl oxidase activity. Now, let's switch to part 2. Elastin has a rubber-like property. When you release it, the protein recoils. When you pull it, it stretches. The primary structure of elastin features small non-polar amino acids, also rich in prolyl and lysine, but very little hydroxyprolyl or hydroxylation. As a result, there's very little alpha helix. Similar to collagen, there's a cross-linking between L-lysine and lysine. And this reaction also depends on lysyl oxidase. Similar to other secreted proteins, synthesis of elastin goes through Golgi before being secreted. After secretion, it is called tropoelastin. Tropoelastin combined with other microfibrillar proteins such as fibrillin-1. Please note, fibrillin is a gene associated with Marfan syndrome. Tropoelastin molecules cross-linked to form elastin. Similar to collagen, elastin also uses L-lysine for cross-linking, and L-lysine is generated from lysine by lysyl oxidase. But different from collagen, 3 L-lysine residues and 1 lysine residue are cross-linked into desmosine. Desmosine accounts for elasticity in elastin. In the lung, elastin is constantly turned over. Elastin is degraded by neutrophil elastase. However, elastin degradation needs to be controlled. Key regulator is alpha-1 antitrypsin, a protease inhibitor produced by the liver. Individuals who are homozygous for alpha-1 antitrypsin have high activity of elastase, which causes excessive degradation of elastin, leading to emphysema. Marfan syndrome features elongation of the bones, dislocation of the lens. It is caused by mutation in fibrillin-1, a microfibrillar protein. Now, let's go to part 3. Keratin features cysteine and disulfide bonds. Keratin protein form a alpha helix. 2 keratin chains form coil-coil. 2 coil-coils form protofilament. 2 or more protofilaments form a protofibril. Protofibrils, together with cells, form a hair. Because disulfide bonds present in keratin can be modified using some chemicals, hair can be easily bent to make it curly or straight. Mutation in keratin 5 or 14 cause epidermolysis bullosa simplex. The condition features blisters on the skin.